Hello everyone, and welcome to today's live broadcast, Exploring the Art Behind Protein Transfer. I'm Julianne Chiat of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. Thermo Fisher Scientific is the world leader in serving science. Our mission is to enable our customers to make the world healthier, cleaner, and safer. We help our customers accelerate life science research, solve complex analytical challenges, improve patient diagnostics, and increase laboratory productivity through our premier brands, Thermo Fisher Scientific, Applied Biosystems, Introgen, Fisher Scientific, and Unity Lab Services. We offer an unmatched combination of innovative technologies, purchasing convenience and comprehensive support. For more information, please visit www.thermofisher.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them into the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of the presentation window, or submit your problem through the green Q&A button on the lower left. I would now like to introduce today's speaker, Emily Goplen. Emily Goplen is a product manager for protein detection regions and systems in the Biosciences Division at Thermo Fisher Scientific. She joined the team in early 2015, coming from an academic background in biological sciences and a product management career in the healthcare industry. She currently manages a range of products, including Western blotting regions and kits, protein quantitation assays, and assay development regions. I will now turn it over to Emily Goplen for her presentation.
Thank you so much for that introduction, and good morning to everyone who's joining today. Again, my name is Emily Goplen, and I'm the product manager of the protein detection portfolio here at Thermo Fisher Scientific. Today, we'll be discussing the various technologies used for protein transfer in Western blotting. As a general overview of what we'll be discussing today, we'll start with the general definition of protein transfer and see how it fits within the Western blotting workflow. We'll talk through the factors to consider before choosing which method of protein transfer is best to use and go through each technique and available system. We will wrap up the presentation with post-transfer considerations and tips and tricks for successful Western blotting transfers. Western blotting. Western blotting is a widely used and highly trusted and established method used to detect the specific and targeted proteins in the sample. It begins by using gel electrophoresis to separate the proteins based on their molecular weights. Post-electrophoresis, the proteins are transferred or blotted from the gel matrix to a membrane upon which they are probed with antibodies and detected via chromogenic, colorimetric, or fluorescent detection reagents. The end result is a detectable signal that captures the protein of interest. The transfer step of the Western workflow is integral to the success and outcome of the detection of the target protein. Over 13 million gels are run each year in the US and Europe alone, and over 75% of these gels are used in Western blotting applications. The most traditional form of transfer is done in a wet tank environment. However, over the most recent years, significant investments have been placed to improve the time and maintain the effectiveness of the transfer step. When choosing the right transfer system, there are many considerations to contemplate. Some of these decisions include the convenience of a pre-made stack versus the flexibility of assembling one's own stack, materials needed such as buffers and membranes, and the speed and time impact of the transfer itself. At a high level, the first factor is between traditional wet tank transfer or the use of a fast transfer device. Various devices have been introduced over the years that offer a reduction in total transfer time. Available consumables of pre-made stacks can additionally improve consistency and throughput of your Western blots. However, there is a price associated with fast dry or semi-dry transfer, and this time savings is key to determine whether this is significant to your lab. The next component to evaluate is the type of membrane in which you use. Nitrocellulose and PVDF are the two most commonly used membranes. These membranes are typically available in either sheets or rolls for wet and semi-dry transfers, or within pre-made stacks, offering the very highest level of convenience. Nitrocellulose and PVDF membranes commonly have a thickness of 100 micrometers with typical pore size of 0.2 or 0.45 micrometers. Most proteins can successfully be blotted using a 0.45 pore size membrane, while 0.2 or even 0.1 pore size membranes can be recommended for low molecular weight proteins or peptides less than 20 kilodaltons. Filter paper or blotting paper and transfer buffers are additional components needed to facilitate the transfer from gel to membrane. Traditional transfer buffers contain hazardous material of methanol. However, new fast transfer systems can include a methanol-free transfer buffer, such as the one-step transfer buffer used in the fast semi-dry transfer. The most commonly used transfer technology is electroblotting, which uses an electric current to pull the proteins from the page gel onto the membrane. This method is used due to the efficiency, speed, and quantitative aspects. In general, there are two primary types of electroblotting: traditional wet tank transfer and rapid dry or semi-dry transfer. The first method, wet tank transfer, uses an external power supply and is run at 4 degrees C or often on ice to keep the temperature cooled over the 1 hour to overnight time period. The latter method utilizes a device with internal power supply and can transfer the total protein in less than 10 minutes. The transfer buffer is incorporated into the transfer stacks for the dry transfer system, making this a bufferless system. Nitrocellulose and PVDF membranes are used due to their large surface area to volume ratio. Each of these, however, has various features that provide different benefits to your system. PVDF membranes have a high binding affinity for proteins and nucleic acids, but they must be pre-wetted with methanol or ethanol prior to submerging into the transfer buffer. These membranes also provide better retention of absorbed proteins because of the greater hydrophobicity and are less brittle than nitrocellulose membranes. Conversely, nitrocellulose membranes are a popular matrix because of their high protein binding affinity and their compatibility with various detection methods. Nitrocellulose membranes do not need the additional pre-wetting of methanol or ethanol prior to use in the transfer buffer. However, these membranes can be fragile when dry and more prone to breaking. Let's talk about traditional wet tank transfer. Wet tank transfer involves a series of steps to set up prior to running the gel to membrane transfer. The first step seen here is to prepare the transfer buffer. Tris-glycine buffer with methanol is the most commonly used choice for wet transfer. The buffer should be stored and used at 4 degrees C. Next, it's important to equilibrate the gel and membrane by placing both in a tray containing 25 to 50 ml of the transfer buffer for roughly 10 to 15 minutes with gentle agitation. Next is the assembly of the gel membrane sandwich. Wet 2 sheets of filter paper and transfer the membrane and transfer cassette pads with the transfer buffer by submerging them briefly. When using your PVDF membrane, it's important to submerge that membrane in 100% methanol for about 30 seconds prior to placing it into the transfer buffer. The gel must be on the cathode side relative to the membrane. When assembling the gel sandwich, it's important to avoid the air bubbles between any of the layers, as this can result in incomplete or uneven transfer. Load the cassette and connect it to an external power supply. Small molecular weight proteins tend to pass through the membrane more readily and may require shorter transfer times. Apply the appropriate transfer time and power setting to run the transfer. Wet tank transfer is the most commonly used transfer method. However, it does require additional setup time and the use of a methanol-based transfer buffer, which is a toxic and hazardous material needing special disposal. When performing a wet transfer, gels are electrotransferred at either a constant current or constant voltage from as little as 1 hour up to overnight. Transfers are typically performed at 4 degrees C to mitigate the heat produced and may be run in an ice pack for further cooling. The transfer efficiency improves with increased transfer time and is better and often needed for high molecular weight proteins. With increasing time, there is a risk of over-transfer of the proteins through the membrane, causing stripping or blowing out. This can be seen in particular with low molecular weight proteins when using membranes with a larger pore size. For this reason, smaller pore size membranes are often used with low molecular weight proteins. The primary issue with wet tank transfer is the use and handling of hazardous methanol-based transfer buffer. To combat this issue, Invitrogen mini-blot module was designed to use 2 to 4 times less methanol-based transfer buffer than other comparable transfer modules. Just imagine handling less and spilling less of that methanol-based transfer. The leak-free and robust device incorporates a fitted gasket for improved sealing. The robust electrode can optimize protein transfer in as little as 30 to 60 minutes. Semi-dry blotting. For semi-dry protein transfer, the transfer sandwich is placed horizontally between 2 plate electrodes in a semi-dry transfer apparatus. The speed of the transfer is improved by maximizing the current passing through the gel instead of around the gel as seen in wet tank transfer. To accomplish this, the amount of buffer available during the transfer is limited to that which is contained in the sandwich. The filter paper used should be cut to fit the exact dimensions of the gel, and it's often beneficial to use extra thick filter paper for high molecular weights, as it can hold more transfer buffer. The length of time that traditional wet tank transfer can require is greatly reduced by the use of a rapid semi-dry transfer device. The Pierce Power Blotter incorporates the ability to transfer 1 to 2 mini gels or 1 to 4 mini gels on a single cassette. Use of the one-step transfer buffer also reduces the transfer time down to less than 10 minutes and eliminates the use of any methanol-based transfer buffers. The system also uses an integrated power supply and reduces the need to hook up to an external power supply. This produces high-efficiency protein transfer across a broad range of protein sizes. Data seen here shows the protein transfer efficiency of tank transfer over 16 hours compared to the Pierce Power Blotter in 10 minutes. HA-tagged protein expressed in E. coli was purified using anti-HA magnetic beads. Samples were prepared and run in an SDS gel, subsequently transferred to a nitrocellulose membrane. Efficiency of the tank transfer with use of Tobin buffer over 16 hours is compared to the Pierce Power Blotter using the one-step transfer buffer in 10 minutes. Membranes were probed with anti-HA antibody, washed and probed with goat anti-mouse HRP, washed, and finally incubated in the SuperSignal West femto chemiluminescent substrate. Here you can see the image captured showing the comparison of these two systems. The image was captured on the thermoscientific MySCL imager, and band densitometry was determined and analyzed using the MySCL analysis software. Results from the experiment show an increased intensity per well up to 2 times from the lysate and at all anti-HA magnetic bead volumes. The Pierce Power Blotter is a fast, efficient transfer device that offers a great reduction in time compared to wet tank transfer. The other benefit of this system is its flexible and open system, allowing for the high throughput of 4 mini blots or 2 midi blots. High protein transfer efficiency can be realized over a variety of protein sizes with the added convenience of a pre-programmed method to run either low, medium, or high molecular weights, in addition to creating customized proteins. Dry transfer. Dry electroblotting provides the highest level of convenience in fast transfer devices. The buffer ion reservoirs are incorporated into the gel matrix or the transfer stacks instead of a buffer tank or soak papers. The high ionic density of the gel matrix enables the rapid protein transfer and provides consistency and control. A reduction in preparation time, run time, and cleanup time is the ultimate benefit of the dry system. Wet and semi-dry protein transfer methods require the setup of the gel membrane stack and cleanup of either the cassette or the transfer tanks, adding time and possible variability to the transfer stage. The Invitrogen iBlot 2 system provides reduced hands-on time and improves the user efficiency that allows for a high throughput of gel to membrane transfers. The pre-made consumable stacks are designed to be intuitive, simple, and convenient, and leaves no liquid cleanup to post-transfer. The system is similar to the semi-dry device in offering both pre-programmed settings to simplify the transfer method or allowing custom programs to fit any unique need. Data shown here shows the strong performance of the iBlot 2 system regardless of gel chemistry. Serial dilutions of phosphorylated EGF receptor was run on 3 different gel types: the Bolt 4 to 12% bis-triz, New Page 4 to 12% bis-triz, and 4 to 20% trizglycine. The proteins were then transferred using the iBlot 2 device and transferred to either PVDF or nitrocellulose membrane stacks. Post-transfer, these membranes were probed with phospho-EGF mouse antibody, washed, and then probed with anti-mouse HRP. These blots were incubated in the Novac CCL chemiluminescent substrate, and high performance can be visualized across the 6 systems. Additionally, transfer efficiency followed by the NIR detection of fluorescently labeled secondaries on both nitrocellulose and PVDF membranes in the 700 and 800 NIR channels can be visualized post-use of the iBlot 2 device. Additionally, we show the combination of the 700 and 800 channel with an overlay of these 2 targets. Clear and sharp bands of both the protein ladder and the target protein are detected after this dry system transfer. They are easily distinguished using the fluorescent detection systems, and crisp bands are detected. Crisp bands are important for detection, and iBlot 2 cleanly transfers without the blowout or smudging. The iBlot 2 blotting device provides fast, reliable protein transfer efficiency in a user-friendly and highly convenient format. It is a buffer-free system that also offers ready-to-use stacks in both mini and midi-sized gels, and both for nitrocellulose or PVDF membranes. The system is capable of high turnover and throughput due to the easily and non-cleanup setup of the iBlot 2 device. It also has the ability to run 1 midi or 2 mini gels per transfer. When evaluating what system is most beneficial to you, it's important to weigh the differences between each method. This includes the convenience, time, throughput, and price. The first difference is in convenience. Convenience and high throughput capacity are benefits of the iBlot 2 and Power Blotter devices. However, the iBlot 2 can transfer only 1 midi or 2 mini-sized gels, while the Power Blotter can transfer double that capacity on a single cassette. Wet tank transfer allows larger amounts of methanol-based transfer buffer to be used, but can offer flexibility on the number of gels run per transfer. The iBlot 2 provides a buffer-free setup and no cleanup. The Power Blotter can double the capacity of the iBlot 2 device, but does require the use of the one-step transfer buffer for rapid transfer. The second difference is in the use of consumables. The iBlot 2 offers pre-made transfer stacks, which can offer consistency and less hands-on time. No more building your own transfer sandwich. The Pierce Power Blotter and Wet Tank Transfer require a build-your-own stack setup, but they do have the ability to use different buffers and stack materials to fit an individual's needs. Price is always a consideration as well, and the last consideration here. With high convenience comes a higher price, and this convenience resonates with many high throughput labs in its ability to speed up time and increase reproducibility within the lab. For cost-conscious labs, however, the open system of the Power Blotter or a wet tank transfer may continue to be the most appropriate option. The value for time continuum can be visualized here when it comes to the comparison of wet, semi-dry, rapid semi-dry, and dry blotting systems. Traditional tank transfer can take up to 1 hour to overnight, run at 4 degrees C or on an ice pack to reduce the heat. The rapid semi-dry or dry transfer systems offer transfer in less than 10 minutes. The total time compared between the dry transfer system, rapid semi-dry, and wet transfer can be visualized here. Again, with less time comes a greater cost in the amount of the consumables and the need for a device to be run. For ultimate convenience and a time reduction, the iBlot 2 dry system is most appropriate. Post-transfer, many times it is desired to check the efficiency of the transfer. Staining the gel post-transfer is often used as a way to confirm the complete transfer of proteins. However, staining the gel is unreliable as it does not indicate how well the proteins transferred to the membrane. A more effective way to monitor transfer efficiency is to stain the membrane with a reversible stain. Ponceau S is a widely used stain. However, it has limited sensitivity and can fade quickly, making it difficult to document. The Pierce reversible protein stain from Thermo Fisher Scientific is more sensitive, easier to document, and is permanent until it's removed. It also has a lower limit of detection, over 5 times more sensitive than the traditional Ponceau S stain. The Pierce reversible stain is safe, simple, and has a quick protocol that results in a turquoise blue band in less than 5 minutes. The stain can be removed in under 15 minutes, and further, can be stored at room temperature, saving precious refrigerator space and eliminating the need to equilibrate prior to use. The bottom right of this slide shows you a comparison of the Pierce reversible stain and the Ponceau S stain. The data here shows the benefits of the Pierce reversible stain over the traditional Ponceau S. Data to the right shows the comparison of these stains on PVDF membrane. The Pierce unstained protein molecular weight marker was serial diluted and run in lanes 1 through 9. The 4 to 20% tris-glycine SDS page gel was run and followed by blotting to a PVDF membrane. Blot A was stained with the Pierce reversible stain, and Blot B was stained with the Ponceau S. You can see significantly better staining with the Pierce reversible stain over the Ponceau S. High background is one of the most common issues scientists face post-transfer during Western blot detection. Background can be seen regardless of the protein transfer method, and is primarily due to poor quality primary antibodies. Before blocking, reagents such as the SuperSignal Western blot enhancer can be used to improve the signal and reduce the background of your Western blot. Using the SuperSignal Western blot enhancer can allow you to achieve 3 to 10-fold better signal intensity and sensitivity compared to detection performed without this reagent. For difficult to detect proteins or low affinity antibodies, the SuperSignal Western blot enhancer reagent can improve the signal-to-noise ratio and enhance your specificity. It does this in 2 ways. First, the enhancer helps to expose the epitopes in order to enable better binding. It also promotes specific binding to the target protein by increasing the ionic strength so that non-specific binding falls out. Even with high promiscuous antibodies that bind to anything, you can get great results using this product. The data shown here uses cell lysates loaded and run on SDS page gels at 1.25, 2.5, 5, and 10 micrograms per lane, transferred to a nitrocellulose membrane and probed using protocols both with and without the use of SuperSignal Western blot enhancer. Here on the left, we can see SuperSignal Western blot enhancer helps to enable the detection of very low abundant targets. On the right, we see a similar effect using the SuperSignal Western blot enhancer in its ability to improve the lower limit of detection for the chemiluminescent substrate. Protein transfer tips and tricks. The overall transfer efficiency and variability in electroblotting methods is dependent upon the protein size and charge, gel type, membrane type, transfer buffer composition, time, transfer temperature, number of gels, and the list goes on. When a protein transfer does not work to the desired standard, there can be many different factors at play. Some helpful tips to help get the desired results that you want is to look at your SDS and methanol and decide to use high-grade methanol and SDS. Low-quality SDS or methanol can generate background and increase conductivity, resulting in poor protein transfer. SDS coats the protein to allow transfer. Increasing your SDS for large molecular weight proteins and removing SDS for small molecular weight proteins can help with the transfer. Methanol removes residual SDS from proteins. Decreasing the amount of methanol to less than 10% for large molecular weight proteins and increasing for small molecular weight proteins can help achieve better transfer. When determining the protein transfer efficiency of semi-dry or dry protein transfer, regardless of the technique used, some proteins may remain in the gel post-transfer. Gels are often more than 10 times as thick as the membrane and thus can hold large amounts of volume and protein. Complete transfer out of the gel and into the membrane of every protein is not always possible. Wet tank transfer provides a buffer or wet environment system, allowing transfer around the gel. Semi-dry and dry transfer systems force the movement directly through the matrix. Additionally, staining of the gel is not a precise reflection of the transfer efficiency, but rather reversible membrane stain should be used to determine the effectiveness of the protein transfer. When transferring high molecular weight proteins, high or broad-range pre-stained protein ladders provide an inaccurate assessment on the unstained target protein transfer. Covalently attached hydrophobic dyes affect the charge of the recombinant protein of the protein ladder, thereby affecting the binding capacity and transfer efficiency of your pre-stained ladder. This is particularly noticeable in 4 to 20% trisglycine ladders. However, when transferring high molecular weight proteins on a fast transfer device, additional time or voltage may be needed to completely transfer the large molecular weight proteins out of the gel and into the membrane. Despite popular belief, rapid transfer systems can equally be as effective of transferring high molecular weight proteins from the gel to the membrane. This can be seen on the right-hand side of the slide. EGFR, mTOR, and ECM29 all have molecular weights that are greater than 190 kilodaltons. These proteins were transferred with the Pierce Power Blotter and wet tank transfer. Both were evaluated by staining the membranes post-transfer as well as completing the Western blot results. You can see similar, if not better, transfer efficiency using the rapid semi-dry transfer device as opposed to the wet tank transfer. Today we covered many details around the primary transfer methods for Western blotting. Each method has its own benefit, whether it be convenience, consistency, price, or time. It's important to evaluate the need of the individual lab to assess which method is best for your needs. Thermo Fisher Scientific offers solutions and products to fit the needs for customers across many areas of protein biology. At this point, we can open up for any questions.
Great. So thank you for that informative presentation. Before we get started on the question and answer session, I'd like to remind our audience on how to submit questions. You can submit questions by typing them into the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Our first question, for tank transfer, what is better for transfer process, ethanol or methanol?
So either ethanol or methanol can be used in the transfer buffer. Methanol is toxic, so you will need to be careful using it. But analytical grade methanol has to be used due to the impurities that methanol can cause in protein transfer issues. So in effect, either is available to be used, keeping in mind that methanol should be used at a high analytical grade.
How much methanol is used in the transfer buffer?
Typically, you'll see up to 20% of methanol or ethanol used in the transfer buffer. Methanol is used to help with small molecular weight protein transfers. Some devices, though, such as the Power Blotter or iBlot 2 device, allow transfer without the use of hazardous methanol. The one-step transfer buffer used in the Power Blotter is methanol-free and therefore not toxic as methanol is.
Will addition of SDS to the transfer buffer interfere with protein transfer?
SDS is commonly used for helping large molecular weight proteins transfer effectively. You can use up to 0.05% in the buffer to help increase the transfer efficiency for large molecular weight proteins.
Okay. So we do not have time for any more questions. Today's webcast will be available for on-demand viewing until June 28, 2017. I would like to once again thank Emily Goplen for her presentation. Do you have any final comments?
Again, thank you for the time today. Please take full advantage of the resources that Thermo Fisher does provide on transfers, as well as our full Western blotting technical guides. Thank you for the time, and I appreciate the attention.
Great. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward this announcement to your colleagues who may have missed today's live event. See you next time, and goodbye.